Welcome back to the deep dive. Today we're doing something a little bit different. Usually, we take a stack of articles on tech or history or maybe psychology and we try to find the big idea. But today we're looking at, well, it's basically a battle plan.
That is, that's certainly one way to look at it.
I mean, it is though. We have the source text right here. It's chapter 3 of the NCERT biology textbook, The Plant Kingdom.
Right.
And to the untrained eye, or, maybe just the tired eye of a student, it looks like this massive list of Latin names.
Just a lot of bold text and some diagrams of moss.
Exactly. But if you actually read between the lines, this is the history of the greatest invasion our planet has ever seen.
Yeah.
It's the story of how life literally crawled out of the ocean, fought against gravity, learned to survive without water, and eventually covered the entire Earth in forests.
You are setting a very cinematic stage for what is essentially a botany review. But you aren't wrong. The evolutionary transition from algae all the way to angiosperms flowering plants is basically just a series of technological innovations by plants to conquer dry land.
Exactly. And that is our mission today. We are doing a deep dive specifically for you, the learner. The students who are preparing for Neet, or honestly anyone who just wants to wrap their head around the green world. We aren't just going to read the textbook to you.
No, that would be boring. We're going to decode the logic. We really want to find the why behind the what, which is honestly the absolute best way to study. I mean, if you just memorize that pinus has mycorrhiza, you'll forget it in a week.
Right.
But if you understand why a pine tree growing in really poor soil actually needs a fungal partner just to survive, you're going to remember it forever.
So here's the game plan. We are sticking strictly to the NCERT Chapter 3 text, so everything aligns perfectly with your exam prep. We are going to travel up the evolutionary ladder.
Step by step.
Yeah. We'll start in the water with algae, crawl under the wet rocks with bryophytes, get some, plumbing with pteridophytes, and then invent the seed with gymnosperms and angiosperms.
And along the way, we really need to flag the high-yield zones for you. The classification systems, the life cycle dominance shift, and specifically the examples. The examiners absolutely love the exceptions to the rules.
Oh, they do. The gotcha questions.
Oh.
So let's dive right in. Segment 1. I call this the sorting hat. Before we talk about the plants themselves, we really have to talk about how we organize this massive kingdom. The text outlines a history of classification that actually kind of mirrors the history of science itself.
It really does. It moves from very superficial observation to deep, structural, and genetic understanding. We start with what the text calls artificial systems. This is Linnaeus.
Right, the father of taxonomy. But the text calls his system artificial. That feels a bit harsh. What was actually the problem with it?
Well, put yourself in the shoes of an early botanist. You walk into a forest. How do you group things?
By what I can see.
Exactly. You group them by what you can see immediately. Is it a herb? Is it a shrub? Or is it a tree? What shape are the leaves? What color are they? Linnaeus specifically used the structure of the androecium, the male sexual parts of the flower.
Okay, that seems logical enough. If they look alike on the outside, they must be related.
But here is the fatal flaw, and you need to lock this in for the exam. The artificial system gave equal weight to vegetative characters, like leaf shape, and sexual characters.
And why is giving them equal weight a problem?
Because vegetative characters are very easily influenced by the environment. Think about it. If you take a plant and grow it in a flooded swamp, and then you take the exact same species and grow it in a dry desert, its leaves might look completely different.
Oh, I see.
Yeah, the desert one might be smaller, or the leaves might be thicker with a heavy cuticle to save water. If you use Linnaeus' system, you might classify them as two completely different species just because their leaves look different.
Right, so the system was totally unstable. It separated closely related species just because they had a different paint job due to the weather?
Exactly. But sexual characteristics, the flowers, the actual reproductive parts, those are much more conserved. They don't change just because it rained a little more this year. That's why giving equal weight to the vegetative parts was a huge mistake. So science had to move on to natural classification systems.
And the big names to remember here are George Bentham and Joseph Dalton Hooker.
Bentham and Hooker. This system sounds a lot more thorough.
Oh, much more. They didn't just look at the outside. The text says they looked at natural affinities among the organisms. They included external features, but also internal features.
Like ultrastructure.
Yes, what the cells look like under an electron microscope, plus anatomy, embryology, and phytochemistry.
So they are finally popping the hood and checking the engine, not just looking at the color of the car.
Correct. But even the natural system has been surpassed today. Now, we use phylogenetic classification systems.
Philo refers to race or tribe, right? So this is all about evolution.
Yes, this is the modern standard. And the core assumption here, and this is the exact definition you need to know for the test, is that organisms belonging to the same taxa have a common ancestor. We are essentially grouping them based on their family tree.
The text makes a really interesting point here. It says this phylogenetic approach becomes absolutely critical when there is no fossil evidence.
Right, because soft plants don't fossilize well at all.
Yeah.
We don't have a perfect stone record of every moss or algae that ever lived. So how do we know who is related to whom without fossils? We have to use other tools to figure out that common ancestor. The text lists 3 specific modern branches of taxonomy that fill this gap.
Let's run through them because these are very easy points on a match the column question if you just know the basic definitions. Number 1 is numerical taxonomy.
This is the big data approach. In the old days, a human botanist might say, well, I think the shape of the flower is the most important feature. That is totally subjective. Numerical taxonomy removes the human bias. You use computers. You assign a number and a code to all observable characters.
Wait, all of them?
Hundreds of them at once. And the key concept here is that every single character gets equal importance. You feed all this data into the computer, and it spits out the mathematical clusters of similarity. It can process hundreds of characters at the same time.
Okay, that makes sense. Tool number 2 is cytotaxonomy. Cyto means cell.
Specifically, it's looking at the nucleus. This classifies plants based on cytological information, things like chromosome number, chromosome structure, and chromosome behavior. If plant A has 14 chromosomes and plant B has 28, that tells us something very specific about their evolutionary relationship.
And finally, tool number 3, chemotaxonomy.
Which is exactly what it sounds like, using the chemical constituents of the plant to resolve confusion. We are talking about the specific proteins, the DNA sequences, the secondary metabolites they produce.
It's basically CSI botany.
Exactly. Okay, so we have our classification systems down. Now let's actually start the journey. We're going all the way back to the beginning. The water. Segment 2, algae.
The chlorophyll-bearing pioneers.
The text describes them as simple, thalloid, autotrophic, and largely aquatic, found in both fresh and marine water. Let's pause on that word, thalloid. That's a term that keeps coming up.
It just means an undifferentiated plant body, a thallus. It doesn't have true roots, it doesn't have a true stem, and it doesn't have true leaves. It's just a mass of plant tissue.
And the physical variety here is absolutely wild. You have microscopic unicellular forms like chlamydomonas. Then you have volvox, which the text specifically calls colonial.
You have to imagine a hollow, microscopic soccer ball made of thousands of tiny green cells just rolling through the water. That is volvox.
It's amazing. Then you have the thread-like ones, the filamentous forms.
Like Eulophrix and Spirogyra.
Right? And then suddenly you have the massive marine forms, the kelps.
Which can grow to 100 meters tall. They form these massive underwater forests. But biologically speaking, they're still just algae.
Now, for the students listening, this next part is the danger zone. Table 3.1 in the NCERT text. The comparison of the 3 classes of algae. This is where the examiners try to trick you constantly with pigments and storage food.
We really need to go through this methodically. Green, brown, and red. Let's start with Chlorophyceae, the green algae.
Chloro literally means green. They are grass green because they have a dominance of 2 specific pigments, chlorophyll A and chlorophyll B.
Which is incredibly important from an evolutionary standpoint because that is the exact same pigment profile as higher land plants. It's a massive clue that land plants evolved directly from green algae.
Precisely. Now, if you look inside their cells at the chloroplasts, the text lists an entire zoo of shapes. They can be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. But inside those chloroplasts is a very specific, highly testable structure called the pyrenoid.
Pyrenoids, what exactly do they do?
They are storage bodies. And here's the catch. They contain protein besides starch. So green algae store their food primarily as starch, but those pyrenoids have a protein core. Some green algae might also store food as oil droplets, but starch is the main one.
Got it. And they have a rigid cell wall.
Yes, made of an inner layer of cellulose and an outer layer of pectose.
And don't forget the flagella. If they move, how do they move? Green algae have 2 to 8 flagella. They are equal in size and they're apical, meaning they're attached right at the tip of the cell.
Okay, so green algae summary for the notes. Chlorophyll A and B, starch storage in pyrenoids, inner cellulose, outer pectose cell wall, 2 to 8 apical equal flagella.
And the examples you must memorize: Chlamydomonas, Volvox, Eulothrix, Spirogyra, and Chara.
Moving on to class number 2, Phaeophyceae, the brown algae.
These are your marine giants, the kelps, sargassum. They are primarily found in marine habitats. Why are they brown instead of green?
Well, they still have chlorophyll A, but instead of B, they have chlorophyll C, and the green color is masked by carotenoids and xanthophylls. Specifically, you need to know a pigment called fucoxanthin. The text says their color varies from olive green to various shades of brown, depending on the exact amount of fucoxanthin present in them.
Now, what about food storage? They don't just store simple starch like the greens.
No, they store complex carbohydrates. You absolutely need to know these two names, Laminarin or Mannitol.
Laminarin and mannitol, okay? And the cell wall.
It has cellulose, yes. But here is the industrial application that makes them important. The vegetative cells have a cellulosic wall, usually covered on the outside by a gelatinous coating of algin.
Algin, that's a hydrocolloid, right? It holds water. We use it commercially as a thickener.
We do. Structure-wise, these brown algae are more complex than the green ones. The plant body is usually attached to the substratum by a holdfast.
Which acts kind of like a root to anchor them.
Right. Then there's a stalk called the stipe. And finally, a leaf-like photosynthetic organ called the frond.
Holdfast, stipe, frond. But the biggest difference here, and the most common exam trap, is the flagella situation.
Green algae had equal apical flagella. Brown algae are different. They only have 2 flagellas. They are unequal in size, and they are laterally attached. They stick out of the side of the zoospore.
And the spores and gametes themselves are pear-shaped. The text uses the word pyriform.
Correct. Pyriform means pear-shaped. The examples to memorize here are Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus.
Finally, we have the deep dwellers, class Rhodophyceae, the red algae.
The majority of these are marine with greater concentrations found in the warmer areas. And the text notes a really cool ecological fact. They occur in both well-lighted regions close to the surface of water and also at great depths in oceans where relatively little light penetrates.
How do they manage to photosynthesize in the dark down there?
It's all because of that red pigment. It's called R-Phycoerythrin. It is incredibly good at absorbing the blue and green light wavelengths that actually penetrate deep into the water column.
That makes perfect sense. What about their food storage?
They store food as Floridean starch. And the text makes a very specific chemical comparison here that you need to know. Floridean starch is very similar in structure to amylopectin and glycogen.
That is a very specific detail. Write that down. Floridean starch equals amylopectin and glycogen structurally. Now, what about the flagella question for red algae?
This is the easiest one to remember out of the 3 because the answer is 0. Red algae have absolutely no motile stages in their life cycle.
No flagella at all.
None. Their vegetative reproduction is by fragmentation. They reproduce asexually by non-motile spores, and they reproduce sexually by non-motile gametes. They rely entirely on water currents to bring the gametes together for fertilization.
One of the examples here, Polysiphonia, Porphyra, Gracilaria, and Gelidium.
Right. So before we leave algae behind, we have to mention why we should care about them beyond just exam questions. The economic importance.
Besides making the ocean look green and brown and red?
Well, for starters, at least half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. They are the primary producers of energy-rich compounds that form the basis of the food cycles for all aquatic animals.
But specifically for humans.
Many species of Porphyra, Laminaria, and Sargassum are among the 70 species of marine algae used as food.
And we mentioned the hydrocolloids earlier.
Yes. Certain marine brown and red algae produce massive amounts of hydrocolloids. These are water-holding substances. We already mentioned algin from brown algae. There's also carrageenan from red algae. These are used commercially all over the world.
And then there's agar.
Oh, agar is huge. Obtained from Gelidium and Gracilaria. It's one of the commercial products used to grow microbes in petri dishes, and it's also used in preparations of ice creams and jellies.
And we can't forget the space traveler's best friend, chlorella.
Chlorella is a unicellular alga rich in proteins. It's actually used as a food supplement even by space travelers.
All right, I think we've conquered the water. Let's step out of the ocean. We are crawling onto the white rocks now. Segment 3, bryophytes. The text calls them the amphibians of the plant kingdom.
It is a totally perfect analogy.
Yeah.
Think about amphibians like frogs. They can live on land, but they absolutely have to go back to the water to lay their eggs. Bryophytes are exactly the same. They live in the soil, but they are entirely dependent on water for sexual reproduction.
But why? What's the mechanical reason?
Because their male gametes, the antherozoids, have flagella. They're biflagellate. When they're released from the male sex organ, the antheridium, they literally have to swim through a thin film of water to reach the female sex organ, the archegonium. If it's bone dry outside, they can't swim and they can't reproduce.
That explains why you only ever find them in damp, humid, and shaded localities.
Exactly. Now, we hit a major evolutionary concept here, the shift in life cycle dominance. If you look out your window at a tree or a flower, the big green plant you see is the sporophyte. It's diploid. But in bryophytes, it's completely backward. The dominant, independent photosynthetic phase, that green carpet you see covering the rocks, is the gametophyte. It is haploid. It produces gametes.
So if the gametophyte is the main plant, where is the sporophyte?
It's essentially a freeloader. After fertilization, the zygote develops into a multicellular body called a sporophyte. But this sporophyte is not free-living. It remains physically attached to the photosynthetic gametophyte and just sucks nourishment from it.
That is a very distinct evolutionary stage. Let's break down the two main groups of bryophytes the text gives us, liverworts and mosses.
Liverworts are the simpler of the two. Marchantia is the classic textbook example. The plant body is thalloid, and it's dorsiventral, meaning it's closely oppressed to the substrate. It just lies flat on the ground.
And they have a really unique form of asexual reproduction, those little cups.
Gemma cups. This is highly testable. Gemmae are green, multicellular, asexual buds. They develop in small receptacles called gemma cups, located right on the thalli.
How do they disperse?
It's basically a splash dispersal mechanism. When a raindrop hits the cup, the kinetic energy splashes the gemmae out onto the nearby soil. The gemmae become detached from the parent body and germinate to form entirely new individuals.
That's brilliant. Then we move up slightly in complexity to the mosses. Examples here are Funaria, Polytrichum, and Sphagnum. Their life cycle is a bit more intricate. The predominant stage is still the gametophyte, but it consists of two separate stages.
Right. When a moss spore lands on the ground, it doesn't just immediately grow a leaf. First, it germinates and grows into what is called a protonema stage.
Yeah.
You need to know that word. Protonema.
What does that look like?
It's a creeping, green, branched, and frequently filamentous stage. Honestly, if you looked at it under a microscope, it looks just like a green alga.
Which totally makes sense given who their ancestors are.
Exactly. It's evolutionary baggage. Then, from this primary protonema, a lateral bud develops. This grows up to become the second stage, the leafy stage. That is the upright green moss plant that we easily recognize. It consists of upright, slender axes bearing spirally arranged leaves. And this is the stage that actually bears the sex organs.
What about their ecological importance? They seem so small. Why do they matter?
They are small, but they are mighty. Mosses, along with lichens, are the very first organisms to colonize bare rocks. This is massive for ecological succession. They literally secrete acids, decompose the solid rock, and create the first thin layer of soil, making the substrate suitable for the growth of higher plants.
And they prevent erosion too, right?
Yeah, since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion.
And we have to specifically mention sphagnum.
Oh, absolutely. Sphagnum is a moss that provides peat. Peat has been used as a fuel for centuries. But for biology students, the key fact is its water-holding capacity. Because it acts like a sponge, sphagnum is used extensively as packing material for transshipment of living material. If you mail a live plant across the country, its roots are probably wrapped in wet sphagnum moss.
All right, we have a good layer of soil going now. It is time for plants to get taller. Segment 4. Pteridophytes, the ferns and the horsetails.
This is the moment plants finally invented plumbing.
Vascular tissue.
Yes, xylem for water and phloem for food. This changes everything. When you are a moss, you have to stay short because you rely on simple diffusion or capillary action to move water cell by cell. But once you have vascular pipes, you can transport water from deep in the soil all the way up to leaves that are high in the air.
So pteridophytes are the first terrestrial plants to possess vascular tissues?
Exactly. And because of this new technology, we see a massive flip in the life cycle dominance.
Right. In bryophytes, the gametophyte was the boss.
But in pteridophytes, the main plant body is now the sporophyte. The diploid stage is taken over. And this sporophyte is finally differentiated into true root, true stem, and true leaves.
Speaking of leaves, the text makes a point to distinguish between small ones and big ones in this group.
Yeah, you have microphylls, which are small leaves found in general, like Selaginella, and you have macrophylls, which are large leaves found in true ferns.
Now let's look at reproduction here. We don't have seeds yet. We still have spores.
Yes. The dominant sporophyte bears sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, these sporophylls form distinct, compact structures called strobili or cones.
Cones, like a pine cone.
Similar idea, but much more primitive. You see these strobili in Selaginella and Equisetum. Inside the sporangia, spore mother cells undergo meiosis to produce haploid spores.
And when those spores fall to the damp ground, they germinate, but they don't grow into a big fern.
No, they go into the Gamophyte generation. And in pteridophytes, this Gamophyte has a very specific, highly testable name, the prothallus.
The prothallus. Let's describe it.
The text describes the prothallus as a small but multicellular, free-living, mostly photosynthetic thalloid gamophyte. It's usually heart-shaped and tiny, maybe the size of your pinky nail.
But it has a fatal weakness, right?
It does. Even though the big fern plant has vascular tissue and can survive in drier areas, the tiny prothallus does not. It still needs cool, damp, shady places to grow. Because just like the mosses, the male gametes need water to swim to the archegonia for fertilization.
So this limits where they can live.
Exactly. The text explicitly states that because of this specific requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. They haven't conquered the dry desert yet.
Okay, I want to slow down here for a second. There is a phrase in the text under pteridophytes that says precursor to the seed habit. This feels like a major pivotal moment in plant evolution.
You're completely right. This is arguably the most important concept in the entire pteridophyte section for a Neet student. Let's break it down. Look at most ferns. The vast majority of pteridophytes produce all their spores of similar kinds. They are called homosporous.
Simple enough. One type of spore.
But certain genera, specifically Selaginella and Salvinia, produce two different kinds of spores. Macro, meaning large, and micro, meaning small. Plants that do this are known as heterosporous.
Okay, so we have big megaspores and small microspores. Why does that size difference matter so much?
Because it changes what happens to the female gamophyte. The megaspores germinate and give rise to the female gamophyte. But instead of falling off and growing independently on the damp soil like a prothallus, the female gamophyte in these plants is retained on the parent sporophyte for variable periods.
It stays home on the big plant.
It stays home. And fertilization happens right there on the parent plant. The zygote develops into a young embryo completely inside the female gamophyte, which is still attached to the parent sporophyte.
That sounds an awful lot like a pregnancy.
It is the biological equivalent for plants. The parent plant is providing a safe, nourished environment for the developing embryo, rather than just throwing it out into the harsh world. This event, the retention of the female gamophyte with the developing embryo, is what the text calls a precursor to the seed habit.
It's the bridge.
Yes, it is considered a massively important step in evolution. Selaginella didn't quite figure out how to make a full, hard-shelled seed, but it laid all the necessary groundwork.
That is fascinating. Before we move on to actual seeds, let's just do a quick classification check for the pteridophytes. The text gives 4 classes.
Right. Celopsida, example is Celotum. Lycopsida examples are Selaginella and Lycopodium. Sphenopsida example is Equisetum. And Pteropsida examples are Dryopteris, Paris, and Adiantum.
Got it. Now let's finish the job. We want to conquer the dry land completely. We need seeds that can travel, and we absolutely do not want to rely on a puddle of water for fertilization. Segment 5, gymnosperms.
The name gives it away. Gymnos means naked. Sperma means seed.
Why are they naked?
Because the ovules are not enclosed by any ovary wall. They remain totally exposed, both before and after fertilization. In flowering plants, the ovary wall eventually swells up and becomes the fruit. Because gymnosperms have no ovary, they produce no fruit. The seeds are just sitting there naked on the cone scale.
These are the conifers mostly, the pines, the cedars. The text mentions the giant redwood tree, sequoia, as one of the tallest tree species in the world. These plants seem built for really tough, cold, dry conditions.
They absolutely are. They are adapted to tolerate extremes of temperature, humidity, and wind. Just look at the leaves of conifers. They are needle-like.
Which heavily reduces the surface area so they don't lose water.
Exactly. They also have a super thick cuticle, and they have sunken stomata.
Sunken stomata, meaning they hide the pores deep in pits in the leaf so the dry wind doesn't just suck all the moisture out of the plant. That's very smart.
Extremely smart. Now let's look down at their roots. The roots are generally taproots. But there are 2 specific symbiotic relationships here that examiners test constantly. You need to memorize these.
Relationship number 1, pinus.
Pinus roots have a fungal association. It's called mycorrhiza. The fungus wraps around the roots and dramatically increases the surface area, helping the pine tree absorb water and minerals from really poor soil. In return, the tree gives the fungus sugar from photosynthesis. In fact, a pinus seed actually cannot even germinate and establish itself without the presence of this mycorrhizal fungus.
Wow. And relationship number 2, cycas.
Cycas has specialized small roots called coralloid roots, and these are associated with nitrogen-fixing cyanobacteria.
So cycas has cyanobacteria, coralloid roots. The alliteration there definitely helps for memorization. What about the stems of these plants?
The stems can be unbranched, like you see in cycas, or they can be branched, like you see in pinus and cedrus.
And what about reproduction? Have we finally left the water behind completely?
We have. Gymnosperms are heterosporous, just like Selaginella. They produce haploid microspores and megaspores. The spores are produced within sporangia that are borne on sporophylls, which are arranged spirally along an axis to form lacs or compact strobili or cones.
So we have male cones and female cones.
Yes. The strobili-bearing microsporophylls and microsporangia are called microsporangiate, or male strobili. The microspores develop into a highly reduced male gametophyte generation. And we know this generation by a much more common name.
Pollen.
Exactly. The reduced male gametophyte is the pollen grain. And it does not have a flagella. It does not swim. It flies.
Pollination is completely by air currents, wind.
Right. And here is a crucial difference from the ferns. In gymnosperms, the male and the female gametophytes do not have an independent free-living existence. They remain strictly within the sporangia retained on the sporophyte.
They are completely dependent on the parent tree.
Yes. The pollen grain lands directly on the opening of the ovules borne on the megasporophylls. It sends a pollen tube down, carrying the male gametes straight to the archegonia, and discharges its contents near the mouth. No water required. Following syngamy, the zygote develops into an embryo, and the ovule develops into a seed, a naked seed.
Just a quick note on monoecious versus dioecious here. The text mentions pinus has both male and female cones on the same tree.
Yes, pinus is monoecious, but in cycas, the male cones and the megasporophylls are borne on different trees, so cycas is dioecious.
Good detail. All right, we have reached the climax of the evolutionary story. The plants that essentially took over the world. Segment 6, angiosperms.
The flowering plants, they are the exception to the naked seed rule. In angiosperms, the pollen grains and ovules are developed in highly specialized structures called flowers, and the seeds are enclosed by fruits.
They're divided into 2 massive classes, the dicots and the monocots. What is the quick cheat sheet for telling them apart?
It's all in the name. Dicas have 2 cotyledons in their seeds. They generally have reticulate or net-like venation in their leaves, and their flowers are tetramerous or pentamerous, meaning the floral appendages come in multiples of 4 or 5.
And the monocots?
Monocots have a single cotyledon. They have parallel venation in their leaves. And their flowers are trimerous. The floral parts come in multiples of 3.
That's the external morphology. But the real magic, the defining feature of angiosperms, happens hidden inside the flower during reproduction. Double fertilization.
This is entirely unique to angiosperms. It is an incredibly efficient system. Let's visualize the embryo sac. This is the highly reduced female gametophyte hidden deep inside the ovule, inside the ovary.
By the time it is mature, the embryo sac has 7 cells, but 8 nuclei. Let's list the lineup of those cells.
Okay, at one end, the micropylar end, you have a 3-celled egg apparatus. This consists of 1 single egg cell and 2 supporting cells called synergids.
Then at the opposite end, the chalizal end, you have 3 cells called the antipodal cells.
Right, and finally, smack in the center of the embryo sac is a large central cell containing 2 polar nuclei.
So that's the setup. 1 egg, 2 synergids, 3 antipodals, and 2 polar nuclei in the middle.
Okay, so a pollen grain lands on the stigma, it germinates, and a pollen tube grows all the way down the style, enters the ovule, and goes into the embryo sac. What happens next?
The pollen tube enters one of the synergids, and it releases 2 male gametes. Not just one, it drops off 2.
2 male gametes. Gamete number 1 moves toward the egg cell and fuses with its nucleus. This is the standard fertilization we all know. It's called syngamy. The result is a diploid cell, the zygote. 2N. This zygote will eventually develop into the embryo, the baby plant.
Standard stuff. But what about gamete number 2?
Male gamete number 2 bypasses the egg and moves to the center of the embryo sac. It fuses with those 2 polar nuclei that were just hanging out together. This process is called triple fusion.
Triple fusion because it's 1 haploid male gamete plus 2 haploid polar nuclei. So N plus N plus N. The result is
a triploid nucleus, 3N. This is called the primary endosperm nucleus, or the PEN.
And what does this PEN do?
The PEN develops into the endosperm. The endosperm is essentially the packed lunch for the baby plant. It provides all the nourishment the developing embryo needs while it's trapped inside the seed. Meanwhile, the thynergids and the antipodals just degenerate after fertilization.
So to summarize, syngamy plus triple fusion equals double fertilization, an event absolutely unique to angiosperms.
Exactly. Now consider the why. Why did evolution favor this complicated double fertilization? Let's compare it back to the gymnosperms.
Okay.
In gymnosperms, the endosperm, the food tissue, is actually the haploid female gametophyte, and it forms before fertilization even occurs.
So it's a bit of a gamble.
It's a huge gamble. If the wind blows the wrong way and the pollen never arrives, the pine tree has wasted massive amounts of energy building a food packet for a seed that is never going to exist.
But in angiosperms, because of double fertilization.
Exactly. The endosperm only develops after triple fusion happens, which means it only develops if syngamy also happened. The plant effectively says, I am not putting lunch in the lunchbox until I know for an absolute fact that the kid is actually going to school.
That is incredible efficiency. It saves the plant massive amounts of energy because it never wastes resources on unfertilized ovules.
Which is probably a major reason why angiosperms dominate the planet today. Following double fertilization, the ovules develop into seeds, and the surrounding ovary matures into a fruit that protects those seeds and helps them disperse.
Incredible. Before we wrap this up, we need to clarify one last major concept that ties all of these groups together. Segment 7, Plant Life Cycles and Alternation of Generations.
Right. In plants, both haploid and diploid cells can divide by mitosis. This leads to the formation of different plant bodies, haploid and diploid. The haploid plant body produces gametes by mitosis, so it's the gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. This sporophyte produces haploid spores by meiosis, which germinate back into the gametophyte. This oscillation is alternation of generations.
But the lengths and dominance of these phases vary completely wildly across the plant kingdom. The text gives us 3 distinct patterns: haplontic, diplontic, and haplodiplontic. These terms always confuse students.
It sounds complex, but it's really just about which generation wears the pants. Which one is the main independent photosynthetic plant you see? Let's go through them. Pattern 1 is haplontic.
In the haplontic life cycle, the sporophytic generation is represented only by the one-celled zygote. There are no free-living sporophytes. Meiosis in the zygote results in the formation of haploid spores, which divide to form the gametophyte.
So the dominant photosynthetic free-living phase is the haploid gametophyte. The examples you must know for haplontic are Volvox, Spirogyra, and some species of Chlamydomonas, basically your simple algae.
Pattern 2 is diplontic. This is the exact opposite.
Yes. Here, the diploid sporophyte is the dominant photosynthetic independent phase of the plant. The ganophytic phase is represented by the single to few-celled haploid gametophyte.
The examples here are all the seed-bearing plants, gymnosperms and angiosperms. But here is the massive trap for the exam. There is an algal exception.
Oh, yes. The brown alga Fucus. Fucus is an alga, but its life cycle is diplontic. Write that in big red letters. Fucus equals diplontic.
Got it. And finally, pattern 3 is haplodiplontic.
This is the intermediate condition. Both phases are multicellular and often free-living.
Bryophytes and pteridophytes exhibit this pattern. In bryophytes, the dominant phase is the thalloid gametophyte, and the short-lived multicellular sporophyte is attached to it. In pteridophytes, the dominant phase is the diploid sporophyte, and the gametophyte is the short-lived independent prothallus. But both generations are multicellular.
Right, but again, examiners love the exceptions. While most algae are haplontic, some of them actually show this haplodiplontic pattern.
Which ones?
Ectocarpus, Polysiphonia, and the massive kelps. They are all haplodiplontic.
So, just to review the nightmare match the column, algae are usually haplontic, but Fucus is diplontic, and Ectocarpus, Polysiphonia, and kelps are haplodiplontic.
Which is exactly why you need to review that specific paragraph twice before you walk into the exam hall.
Well, we have covered an immense amount of ground today. From the tiny, slimy Volvox colony rolling through a pond to the giant redwood towering over a forest.
It is an incredible journey of adaptation, from being totally dependent on standing water for fertilization, to utilizing the wind and insects to carry pollen across miles of dry land, from leaving unprotected spores on damp soil to carefully tucking embryos inside tough seeds, complete with their own endosperm food supply, safely hidden inside a fleshy fruit.
It really changes how you look at the natural world around you. The next time you see a small fern growing in the shade of a wall, you can realize that it is essentially a relic, a living fossil from a time before flowers even existed on Earth.
And when you eat a peanut, You're actually eating a dicot embryo that was nourished by a triploid endosperm, all resulting from the highly efficient process of double fertilization.
Bon appetit, I suppose. That is going to be it for this deep dive into the plant kingdom. Good luck with your exam prep, everyone. Focus on those life cycles, memorize those exceptions, and you will do great.
Keep questioning the world around you.
See you next time.